The purpose of this video is to give my biology students an opportunity to see the types of questions that they're going to have on their final. This represents every single question that you will see on your final. And of course, because this is a video and you have the ability to pause, I will be going faster than I will be normally in class when we're playing the game. All right, we're going to do one column at a time. Here we go. What is the correct order of the scientific method? Hint, there are 5 steps. Give you about 45 seconds for this one. Also, I am eating my lunch, so I beg your pardon if you hear me munching away. 5, 4, 3, 2, and 1. Got to identify the problem, form a hypothesis, perform the experiment, analyze your data, and then you modify the experiment or you communicate results. Also, if you want an extra time to look these up in your study guide or to just try to figure out what they are, remember to pause the video. All right, convert 7.32 kilometers to meters and convert 7.7463 times 10 to the third into normal notation. I'll give you about 30 seconds for this one. All right, 15 more seconds. Again, if you want a little bit more time to figure this out, pause the video when I start counting down from 5, 4, 3, 2, and 1. Okay, so kilometers is bigger than meters. Therefore, my decimal is going to be moving to the right, and it is moving to the right 3 spaces. 1, 2, 3. You add a 0. And 10 to the third, any positive exponent means the number is going to get bigger. That means I move the decimal 1, 2, 3 spaces to the right. If this is a negative exponent, the decimal is going to move to the left. The process used to find answers to questions is called what? What's the correct order of the sections in a lab report and data with units, numbers that is measured is blank data? About 40 seconds for this one. All right. 5, 4, 3, 2, and 1. All right, that's a scientific method. Title, objective, hypothesis, procedure, data, and conclusion. Quant, count. Quant, count. Counted, measured data with units is quantitative data. What are the 3 goals of science? The variable that is deliberately changed is called what variable? About 40 seconds. And I apologize. I am at school. That's not normally where I record my videos. And of course, you're going to hear noises from other classrooms. Like I said, all right, the answer is coming in 5, 4, 3, 2, and 1. Okay, it's to investigate and understand the natural world, to explain events, in the natural world to use derived explanations to make useful predictions, and the manipulated or independent variable is the one that is deliberately changed. Easiest way to remember is the independent is the one that I change. All right, on to organic molecules, chapter 2. What are the 3 particles that make up an atom? All right, answer in 5, 4, 3, 2, and 1. Protons, electrons, and neutrons. What are all of the elements of life found in carbohydrates, lipids, proteins, and nucleic acids? You'll find this in the 2-3 class notes. All right, answers coming in 5, 4, 3, 2, and 1. Hydrogen, carbon, oxygen, nitrogen. Nitrogen is primarily found in nucleic acids and proteins. Phosphorus, nucleic acids. Sulfur, nucleic acids and proteins. Hydrogen, carbon, and oxygen primarily are carbohydrates and lipids. And then carbohydrates are the immediate source of energy. Which organic molecule stores genetic information as a genetic code? What are the monomers of proteins? And what type of organic molecule is this? All right, answers coming in 5, 4, 3, 2, and 1. All right, nucleic acids store genetic information. Remember, DNA gives us our characteristics. Messenger RNA helps DNA make proteins. Proteins give us our characteristics. Monomers of proteins, amino acids, and this is a lipid. This is glycerol with 3 fatty acids. Glycerol and fatty acids are the monomers of lipids. Glucose and fructose are both examples of what type of organic molecule? Sketch the structure of a carbohydrate. Phosphate groups guanine, cytosine, and adenine are found in both blank and blank. Which scientist took the famous Photo 51, which helped determine DNA's structure?
Hey, you're good. I've got a mask on mine. Oh, could I borrow your
I'm recording a video, but yes. Go ahead. That's right.
Okay, can I borrow your box?
Mm-hmm. It's just my Jeopardy. I always put it in Google Classroom so they can look at the questions the night before the test. Answers coming in 5, 4, 3, 2, and 1. All right, so carbohydrates are glucose and fructose. Of course, phosphate groups guanine, cytosine, and adenine are found in both DNA and RNA. Note, uracil is RNA, thymine is DNA. Sugars are different too. This is what a carbohydrate looks like, and Rosalind Franklin took the famous Photo 51. All right, on to energy and climate. Sketch a food chain with 1 producer and 3 consumers. All right, answers coming in 5, 4, 3, 2, and 1. Okay, there are lots of possible answers, but you have to start with a producer. Okay, it could be grass, could be wheat, could be corn, could be algae, could be anything that uses photosynthesis. Primary consumer eats the producer, secondary consumer eats the primary consumer, and the tertiary or the third consumer eats the secondary consumer. And of course, there are lots of examples for that. All right, an herbivore is any organism that eats a blank. What natural process releases carbon dioxide from living things into the atmosphere? All right, answers coming in 5, 4, 3, 2, and 1. All right, so an herbivore, they eat producers, they eat autotrophs. They're the same thing. They eat plants. You're going to have to recognize producers or autotrophs, though. And living things, through the process of getting energy for their cells, it's called cell respiration, they release carbon dioxide and water vapor from that reaction. Blanks help the nitrogen cycle by releasing nitrogen in animal waste. What process speeds up the release of carbon from fossil fuels into the atmosphere? And true or false, matter decreases over time. Answers coming in 5, 4, 3, 2, and 1. All right, decomposers break down organic matter and they release that nitrogen, among other things, back into the soil. Combustion, which is a fancy word for burning, and false. Matter cannot be created or destroyed. It's called the law of conservation of matter. Matter is recycled. Matter is recycled, or it's transformed. The lengths of days differs throughout the year because the blank of the Earth is tilted. What branch of biology deals with the interactions between organisms and their environment? What types of organisms supply food to ecosystems? And only about 10% of the available energy in a lower trophic level gets transferred to the next level. What happens to the other 90%? About 20 seconds. Answer in 5, 4, 3, 2, and 1. Okay, so the axis of the Earth is tilted, and that gives us seasons, and it gives us different times, different lengths of day. That is the biological discipline, ecology. Producers or autotrophs supply food to the ecosystems. They are the base of all food chains, and 90% of that energy is lost as metabolic heat. All right, DNA structure and replication. What organic molecule is that? Answer in 5, 4, 3, 2, 1. That is DNA, or a nucleic acid. And that's what Na stands for. Avery's experiments showed that bacteria are transformed by what? And in eukaryotes, where do you find DNA? Answers coming in 5, 4, 3, 2, and 1. All right, Avery figured out that DNA was the transforming factor, and DNA is found only in the nucleus. It makes messenger RNA in the nucleus, and then messenger RNA leaves to go to the ribosome to make proteins. If DNA strand 1 is AAC-GCT and is being used in replication, the complementary strand will be what? And after blank blank, each new molecule will have one old strand and one new or complementary strand. And I beg your pardon, this is only a 2-point question. Let me change that real quick. Answer coming in 5, 4, 3, 2, 1. All right, so A, T, C, G, G, C, C, G, T, A. And of course, after DNA replication, each molecule is one old or original and one new or complementary. All right, which type of gene mutation is never a frame shift mutation? It should sound familiar because assuming it's 2021 when you're watching this video, this was one of the questions on your Chapter 12 unit test. Answer in 5, 4, 3, 2, and 1. It's a substitution mutation. Transfer RNA, ribosomal RNA, and messenger RNA are all involved in the process of blank blank. Blank molecules are produced during transcription. Answer coming in 5, 4, 3, 2, and 1. Okay, those all three types of RNA are involved in protein synthesis, and RNA, specifically messenger RNA, or mRNA, as we lovingly call her, are produced during transcription. And I'm going to make a quick update. I just caught a mistake. During blank, the cell uses information from messenger RNA to produce proteins. Where does transcription take place? And proteins are made by making long chains of blank blanks. Answer coming in 5, 4, 3, 2, 1. During translation, messenger RNA is used to make proteins. And remember, messenger RNA indirectly is carrying DNA's message. All right, transcription, the making of messenger RNA, happens in the nucleus, and proteins are polymers made of monomers called amino acids. Given the following DNA nucleotide sequence, what are, number 1, the second strand of DNA, number 2, messenger RNA, number 3, transfer RNA, and number 4, amino acids, given TGA, CCT, GAC. And I'm sorry, I'm kind of speeding things up here a little bit. I've got about 4 minutes until the bell rings and about 5 until students start arriving, and I need to make sure this video is finished. Answer coming in 5, 4, 3, 2, 1. All right. DNA 2 is ACTGGA-CTG. Again, you use DNA 1 for messenger RNA, ACU-GGA-CUG. Transfer RNA, you use messenger RNA, that is UGA, CCU-GAC, and you use messenger RNA to get the amino acids, that's threonine, glycine, and leucine. Last column. Solution with a pH above 7 is called a blank. Solution with a pH below 7 is called a blank, and a solution with a pH equal to 7 is called blank. Answer coming in 5, 4, 3, 2, and 1. Above 7, we call that basic or alkaline. Below 7, we call it acidic. And equal to 7, we call neutral. The repeated movement of water between Earth's surface and the atmosphere is called what? Proteins are polymers made from the monomers called blank blanks. This has been in there a couple times, which is just fine. If you know it once, you know it a couple. All right, answer coming in 5, 4, 3, 2, 1. All right, that is the water cycle and amino acids. If an enzyme's activity slows down as temperature increases, what is likely happening to the enzyme? And the original source of almost all of the energy in most ecosystems is what? Answer coming in 5, 4, 3, 2, and 1. All right, so the enzyme's being damaged, destroyed. It's also called being denatured. Remember, adding heat breaks bonds, and proteins and enzymes have very unique, specific three-dimensional shapes. And if those bonds start getting broken and the protein shape gets altered, then the enzyme substrate complex key fit is not going to work, and that will, of course, slow down the activity of the enzymes. And sunlight or solar energy is the source of most of the energy in ecosystems. Last questions. The diagram below shows the relative amount of blank available in an ecosystem. Sugar and phosphate form the blank of DNA. And what types of bonds connect the nucleotides or nitrogen bases in the center of the DNA double helix? All right, I'm going to sound a little muffled because I just put my mask on because we're probably going to have kids here any minute. Answer coming in 5, 4, 3, 2, and 1. Okay, this is showing energy transfer. 100%, remember you lose 90%, that leaves 10, that leaves 1, that leaves 0.1. The sugar, phosphate, backbone, and weak hydrogen bonds connect the nitrogen bases. All right, ladies and gentlemen, I hope you found this helpful, and I will see you in class.